Hello and welcome to Chapter 7, Transport in Plants. So you guys have learned quite a bit about transport in plants during STEM, but we're going to go into a lot more detail. So plants use photosynthesis to convert light energy to chemical energy. And simple organic substances like CO2 and H2O and ions are used in their raw form to produce glucose and other carbohydrates. So how does the plant get the water and the CO2? And does the plant have a circulatory system like we do? So how would these trees lift water against the force of gravity? Okay, so here are your Cambridge objectives. Okay, so we're going to talk about a few different types of plant tissues. We're going to talk about dermal tissue, ground tissue, and vascular tissues. So plants have two separate transport tissues, vascular tissue. Okay, the first is xylem tissue. And this is where water and ions are traveling upwards and they move in one direction. So it's a flow from roots to stems to leaves to flowers to fruits. And then there's phloem tissue. This is where sucrose and other assimilates will travel upwards and downwards. Okay, so xylem is one direction, phloem is two directions. Movement of water in the xylem and phloem is by mass flow. Everything travels in the same direction within each column of the xylem or the phloem, and in the case of the phloem, at one time. It would be either upwards or downwards. So note that neither plant transport system carries O2 or CO2. Okay, so this should be a somewhat familiar diagram to you. So this is the process of photosynthesis, right? The tree is bringing in CO2, expiring O2, and bringing in light and transporting it around the tree. And you can see in the roots, it's pulling up water and minerals, as well as O2, bringing in O2 from the soil and expiring CO2 into the soil. So we're going to dig deeper into this process. Okay, so in both the walls of the tubes of the xylem and the phloem are thickened by the addition of cellulose, and we know that cellulose is an organic compound, it's a polysaccharide, as well as lignin, which is a woody material. And water transport really takes place in 3 steps. So the whole process is called transpiration, and sometimes you'll see it as evapotranspiration. So this is the transport of water and minerals from the roots to the leaves. The 3 basic steps include absorption at the roots, capillary action in the xylem vessels, evaporation at the leaf. Okay, so when you're looking at the xylem, which again is part of the vascular tissue, we are going to talk about the function of the xylem, the types of cells, okay, so this would be vessel elements and tracheids, the direction of movement, as well as transpiration, which is sort of facilitated by the stomata and the guard cells. All right, so stomata, as you should already be aware of, are the pore-like openings that are usually in the bottom side of a leaf. And this is where carbon dioxide enters and while water and oxygen exit. And they're really important in the whole process of transpiration. They're what they allow water and oxygen to escape as a byproduct of photosynthesis. And that's where, to a large extent, they're bringing CO2 in. Okay, so another part of this process is, so that we have the xylem and now the roots. The roots, the different functions of the root vary based on the type of plant and the part that you're looking at the root. So roots have root hairs. Okay, there are single-celled extensions of some cells. They are very thin. A single root can have thousands of root hairs. And what it does is it increases the surface area and absorbs water by osmosis. So if you look at this, you can see the root and then all of the little hairs that are coming out into the soil, okay, and which is obviously increasing surface area and allowing more water to be diffused. Okay, so the water is moving in the cells through osmosis. Okay, and we know that osmosis is the movement of water molecules from an area of high concentration to an area of lower concentration. Okay, and there's the lower solute concentration in the soil, okay, meaning it has the high water potential, and the higher solute concentration in the root is lower water potential. And so remember, guys, this is when you're talking about this in your exam or when you're ever writing about this, this is the type of thing that you're mentioning, that you're talking about. Lower solute concentration yields higher water potential. Okay, higher solute concentration in the root yields lower water potential, which is why water is moving in the direction that it is from the soil into the root. Okay, and there are really two routes for water to move into a root. So there's the apoplast pathway, which is around. Okay, so when water soaks through the cell wall and seeps across the root from cell wall to cell wall and through the spaces between cells. Okay, so then the other is the symplast or the cytoplasm. So this is when water enters cell walls and moves from cell to cell by osmosis. Or through strands of cytoplasm that make direct connection between adjacent cells, the plasmodesmata. Okay, so if you take a look at this model, you can see the root hair and the different paths that water can take. Again, so here's your apoplastic root in pink, and here's your blue in the symplastic root in blue. Okay, and so the big thing here is that in the symplastic, which is like cytoplasmic, you can see it's moving in through the cytoplasm and in through the plasmodesmata between cells. Okay. So when water reaches the stellae, the apoplast pathway becomes blocked, and the endodermis cells have suberin, or in other words, they're waterproof. Take a minute, pause, really look at this video, look at where the arrows are pointing and how the water is moving. Okay, the Casparian strip. This is a belt of waxy material. It allows only minerals in the symplast to pass into the vascular cylinder through the plasma membrane of the endodermal cells. Okay, cells in the vascular cylinder transport water and minerals throughout the plant. Again, this is a really good time to pause and really look at this model. So look at the apoplast and the symplast, okay, and see how they're getting from cell to cell, okay. And here you can see the Casparian strip as the little purple strips that go through. Again, it's waxy material that only allows minerals in the symplast to pass into the vascular cylinder. Okay, another set of models that are helpful. Okay, here is another, this is a very typical sort of Cambridge drawing that you might see. Okay, you can see the endodermis there with the Casparian strips. And this is a scanning electron micrograph, and you can really see all of the details here. So if you look back, you can see the endodermis, which, is endo means inner, and dermis is skin. So you can see it's this under this inner layer here, and you can see the Casparian strips between. Okay. So the xylem, with the long narrow cells, the elements are really, they start as living tissue, so they could be from the cell wall or the nucleus or other parts of the cell. Then they're differentiated into specialized structures and then die. Okay, the xylem is constructed of dead cells that are no longer living; they're just empty shells. Okay, so this is pretty cool if you look at this root cross section. So this one over here is a root cross section, and then on the other side you can see the stem. So here you can see where the phloem is on the root cross section. Okay, and then if you look over at the stem, you can see that the phloem, you can see where the phloem is the top sort of half of the vascular bundle that's there. So the top half is the phloem, and then the bottom half is the xylem. And if you look down to the dicot stem, you can see it better there, where the top sort of half of that vascular bundle is the phloem, and the bottom half is the xylem. Okay, and so looking at a leaf cross-section, you see the different layers, which you guys also learned about in STEM, including the upper epidermis, the palisade parenchyma, the spongy parenchyma, lower epidermis, and then there you can see your stoma. So we're going to learn a little bit more about guard cells. Okay. Again, I encourage you to, at any time, stop and look at these models, go back and really challenge yourself to sort of label what you're looking at. Okay, so palisade and spongy mesophyll cells have really large internal surface, and it's completely for gas exchange. As the carbon dioxide concentration in the air is low, surfaces are larger, are able to absorb more for photosynthesis. Okay, so you do have to know the anatomy of a cross-section of a leaf as well as the root and stem. So if you're looking at this, the upper epidermis, the mesophyll, and the lower epidermis, you have to understand what the stoma is, what air spaces are for, which we'll talk about that in a second. And within the mesophyll, it's important to be able to distinguish between the palisade and the spongy mesophyll. And so let's talk, well, in a second, we'll talk about the stomata. So the stomata are the pores in a leaf, and they're mostly on the undersurface. Each pore is surrounded by a pair of guard cells. So guard cells work to change the shape, which will open or close the stoma. So there are situations where, like if it's really hot outside and there's not a lot of water, it's sort of a water poor condition, the plant will not want a lot of its water to evaporate, and so it will close its stoma more. Okay, so if you see here, you see that the guard cells in this picture are swollen and turgid, okay, and that causes them to bend and the stoma to open. And then guarded cells that are shrunken and flaccid close. Okay. So the air inside leaves is always fully saturated with water vapor. So usually the air outside is less saturated. And so again, a concentration gradient for water vapor exists between the air spaces and the outside. So water will diffuse down the humidity gradient, right, from high to low. The pathway with the least resistance is through the stomata. So how does the water move up? Okay, so what are the water properties? What is capillary action, cohesion, adhesion, tension theory, and bonding?
How do water and minerals get to the leaves? Although roots can exert a slight upward pressure, it is evaporation of water from leaves, a process called transpiration, that moves water molecules and ions up from the roots. Transpiration exerts a pull that is relayed downward along a string of water molecules from leaf to root. Hydrogen bonds cause water molecules to stick together, a phenomenon called cohesion. As each water molecule evaporates, it pulls on the next water molecule, and it pulls on the next. This relays the pull of evaporating water molecules all the way down to the roots. The adhesion of water to walls of the xylem cells helps to keep gravity from pulling the water molecules back down. As each water molecule escapes from the leaf, it pulls a column of water molecules upward. Solutes are transported along with the water. Thus, the plant xylem uses the movement of evaporating water molecules, cohesion, and adhesion to draw water and dissolve minerals from the soil into its roots and upward to its leaves. Hot, dry, windy conditions increase transpiration. If not enough water moves up from the soil to replace the water lost to evaporation, the plant will wilt and it could die. How does a plant prevent excessive water loss? An opening in a leaf is called a stoma. Stomata are generally open during the day to allow carbon dioxide into the leaf, where it is used in photosynthesis. While the stomata are open, water moves out of the leaf by transpiration. This allows the plant to move water and minerals to the leaf, and the evaporation of water also functions to cool the plant. If hot, dry conditions require the plant to conserve water, guard cells close the stomata. This is a trade-off. Although closing the stomata reduces water loss, this also slows down photosynthesis and may cause the plant to overheat.
Transpiration drives the movement of water in plants. So as you saw in the video, the loss of water from leaves by transpiration causes water to travel upwards through the plant by mass flow. And that's a term and a vocabulary term that you need to use, okay? Water travels upwards through the plant by mass flow. The mechanism is called cohesion-tension, and it works like so. So water loss is caused by transpiration. It causes a pulling force, okay? And this is a negative pressure that is produced, and the transpiration is the pull, right? That pulls it up. So water is absorbed by the roots and through osmosis moves from root hair cell to root cortex cell. And it continues to move that way until it gets into the xylem. It moves up the xylem through transpiration. And so this is where water is evaporating from the leaf surface. It is creating this tension and this pull. And through hydrogen bonding, water, which is the cohesion, it creates this, a tension where the water is moving upwards against gravity and out towards those stomatas or stomas. Okay, so the cohesion tension theory. So again, absorption in the root through osmosis. Endodermal cells actively secrete mineral salts, and this is to keep the water potential in the xylem lower, which is really interesting. And this is causing water to be drawn through the endodermis. There's this pulling of water that's caused by cortex cells, which produce a positive hydrostatic pressure inside the xylem. And this is forcing water upwards. Okay. So then the water is moving up through the transpiration pole, which is negative pressure, and the root pressure, which is positive pressure. Okay, so the two important factors here are cohesion, where water molecules tend to stick together through hydrogen bonding, and adhesion, which is water molecules tend to stick to the inside of the xylem. Okay. So the spongy mesophyll cells are not tightly packed together. There are plenty of air spaces that are in direct contact with the air outside the leaf through the stomata. So if air outside the leaf contains less water vapor than inside, right? So it's like more humid inside the plant, then there is a water potential gradient from the air spaces inside the leaf to the outside. Okay, and so again, this is going to cause a pull where water is going to be pulled in one of two ways through that root cortex. Okay, so and you should take a time to try to start quizzing yourself about these layers and which pathway is which. Okay, so the potometer, this measures the water absorption. It's an estimate. You can estimate the rate of transpiration this way. And really it has to be air and water tight. And water is being transpired, entering into the xylem. And you can see here in the calibrated pipette, you can see how much water is being is going down, how much water is being lost to the system. Okay, so factors that affect the rate of transpiration. So light intensity. This affects the opening and the closing of the stomata. Okay, and it is an indirect effect because light intensity will it can raise temperature, it can heat up the plant, and so the stomata, or I'm sorry, the guard cells might close that stomata, okay, which is going to decrease the rate of transpiration. rate of transpiration. That's what it is. Okay, so factors that affect rate of transpiration. One of them is light intensity. So light intensity will affect the opening and closing of the stomata, whether that's a low light intensity or high light intensity will change how much the stomata is going to let moisture out of the plant. So ROT, which stands for rate of transpiration, will potentially increase with the increase of light intensity. So if you look at this graph here, you can see that the rate of transpiration increases steadily, but then at a certain point, with as light intensity increases, it starts to level off, and that's because as if light gets too intense, then the stomata will start to close to prevent too much evaporation occurring, which will then decrease the rate of transpiration, or at least slow it down. Eventually, if light intensity kept increasing, rate of transpiration would start to decrease there. Okay, humidity also affects rate of transpiration. So in an atmosphere that is very humid, right? It contains a lot of water molecules in gas form. There will be a reduction of the water potential gradient between the air spaces in the atmosphere, right? We talked about how there's the water vapor that's moving out of the plant, right, in the air spaces into the atmosphere is going down the concentration gradient. But if the concentration of water outside the plant is increasing, then that's going to change the water potential between the inside, the air spaces of the plant, and outside. And so in this case, the rate of transpiration would decrease. So high humidity decreases rate of transpiration, low humidity increases rate of transpiration. Okay, temperature affects the rate of transpiration. So when temperature increases, we know based on the kinetic model that energy increases. So the rate of diffusion through the stomatopore will increase. Air is able to hold more water molecules at higher temperatures, and so the rate of transpiration increases, right? So more energy, and that's just going to equal more rate of transpiration. Okay, but again, at a certain point, the system would start to break down. So if the plant got too hot, then evaporation would be occurring too quickly, and the plant would start to suffer, and then of course rate of transpiration would decrease at that point. But for the most part, when temperature increases, transpiration increases as well. Okay, wind speed really affects the rate of transpiration. So still air, that is air without wind, makes water molecules accumulate around the stomatopore. Okay, and this reduces water potential gradient and slows the rate of transpiration. Okay, so as water is escaping those pores, they start to accumulate because of cohesion. They start to accumulate to each other around the pores. Which reduces the water potential, right? Because right outside of those stomatas is a higher concentration of water, and so that slows rate of transpiration. But if it's windy, that disperses those water molecules from the stomata, and that increases the gradient in water potential, which increases the rate of transpiration. Okay, so xerophytes are cool. We are surrounded by xerophytes in Arizona. This is a plant that's adapted to live in dry conditions, and there's a variety of adaptations that a xerophyte can have to reduce their loss of water vapor through transpiration. Okay, so one of them is the leaves. So the leaves of xerophytes are very small, and this is to reduce the surface area, whereas in other plants, the leaves are wide and broad to increase surface area. Okay, but in an environment with extremely high temperatures and low water, you want them to be smaller and you want them to be thick to reduce surface area. Okay, so They also have sunken stomata, which is really cool. So if you look at this picture, you can see the stomata that are down here at the bottom of these pores. They're really sunken into the plant, and so this is going to help water to not instantly evaporate the moment that it reaches that stomata. So they're set deep inside so that they're at the base, so that there's a base of a depression that's full of water vapor. And some plants will open their stomata at night to store and absorb CO2, which is also a really interesting xerophyte adaptation. Okay, they have thick, waxy cuticles, and this reduces water loss through the epidermis. Okay, so you can see in a regular plant, the leaves are not, their cuticles are not going to be that thick. Rolling up of leaves, okay, so the lower surface faces inside and traps humid air next to the stomata. And this, of course, is going to vary with conditions. They have leaf hairs, which are interesting. So these will trap damp air, and this helps to reduce air movement, which we learned that air movement or wind increases rate of transpiration. But in a plant that's trying to constantly decrease their rate of transpiration, having these like places, these cavities where air movement is decreased and damp air can maintain, this will cut down transpiration. Okay, so transport in the phloem. So most photosynthesis occurs in the leaves. The reactions take place in chloroplasts, which you know. The compounds that the plant makes are called
in their leaves. These nutrients must then be transported to the rest of the shoot or to the root tips where growth occurs. The leaves are referred to as the source, and the shoot and root tips are referred to as the sink. A source is an organ that produces more sugars than it requires, and a sink is an organ that consumes sugars for its own growth and storage. There are two kinds of vascular conducting tissues in plants: xylem, which conducts water and dissolved minerals, and phloem, which conducts carbohydrates, mainly sucrose. The transport of sugars throughout the plant, called translocation, is carried out by phloem cells. Phloem cells are connected to one another end to end by sieve plates. These are perforated structures that create a direct connection between the cytoplasm of connected phloem cells. Phloem loading is the process whereby carbohydrates enter the sieve tubes at the source. As a result of phloem loading, a high concentration of sugar develops in phloem cells near the source. Phloem loading results in a lowered water potential compared with adjacent xylem cells, causing water to move from xylem to phloem by osmosis. This influx of water creates a high turgor pressure near the source and lower turgor pressure near the sink, causing the movement of water and sugar from the source to the sink. Sucrose is removed at the sink by various active transport mechanisms depending on the cell type. In some plant cells, a transport protein located in the membrane of a storage vacuole called a tonoplast transports sucrose from the sink cell's cytoplasm into the vacuole at the expense of ATP. This reduces the concentration of sucrose in the sink cell, which enhances the movement of sucrose from the phloem cells through the companion cells and into the sink cell. All these events produce a continuous flow of water from xylem to phloem and then back to xylem. Water leaving phloem in the root moves upward primarily due to transpiration, not due to water potential differences between xylem and...
Okay, so many of these are exported from the leaves to the rest of the plant in the phloem. Okay, so when you're looking at the xylem vessel next to the phloem vessel, the xylem vessel carries water one way only, and it's water and dissolved minerals. And there are no end walls between the cells, right? It's just a continuous pipeline. And the thick walls are stiffened with linen, and the vessel itself is dead, right? It's made of dead cells. Where the phloem vessel is made of living cells, and the cells are end to end with perforated walls called sieve plates that are in between each cell. And there's a two-way flow in the phloem vessel, which carries water and food like sucrose. Okay, so let's talk about sources and sinks. The transport of these assimilants is called translocation, okay, and that means from place to place. The assimilants are loaded in the phloem in leaves, and they're often called sources. So the place where the assimilants are loaded are called sources, and they're transported to other parts of the plants that need the assimilants or the food or the sucrose, such as the roots or stems, flowers, fruits, seeds, etc. And those places are called sinks. So the places where the sucrose is being loaded is called source cells, and the places where it's being taken are called sinks. Okay. So let's talk about sources and sinks in more detail and the process by which this occurs. So if you're looking at this model, you can see at the top here, you have your source cell, okay, here. And your source cell is where, oftentimes, most of the time, is where photosynthesis is happening. And sucrose is combined from glucose and fructose, right? So the sucrose is a stable molecule to travel through the plant. And so sucrose is being loaded, okay, from the source cell into the companion cell. So remember, this companion cell is part of the phloem, and it is the intermediary between the phloem and the source cell. So the companion cell is where sucrose is being taken from the source cell and put into the phloem for transport. Okay, so it gets put into the phloem, and at that point, the solute concentration increases, right, making it a concentration gradient shift. Okay, so now there's a lower water potential. So water is moving by diffusion or by osmosis from the xylem to the phloem at this point, right? Because there's a lower concentration gradient of water on this side. So water moves from the xylem into the phloem, and in doing so, it creates this pressure, right? Because now there's more fluid in the phloem than there was originally, on top of there being now sucrose that has been loaded from that source cell. So there's this combination of water and sucrose, which is sap. Okay, right? It's sap. So the sap, there's this pressure. The sap is moving down, okay, because that water is added in there, and that pressure is pushing the sucrose and the sap down. And then once it gets down to another companion cell where it's ready to be loaded into a sink cell, so in this case this is a root cell, but it could easily be this cell going into a fruit. This companion cell starts to unload sucrose from the phloem into the sink cell. So in the process of doing this, now the concentration gradient is changing again, right? Because the solute is decreasing. And so then the water potential is changing. Water starts to move again down the concentration gradient back into the xylem. Okay, and then it starts the whole process over again. And this is the process of translocation, right? Moving food or assimilants, okay, sucrose, from one place, the source where they're created and stored, into sink cells where they're being used for growth or storage or a few other purposes. Okay, so the transport, again, is called translocation. Sucrose and other assimilants travel throughout a plant in these sieve tubes. Okay, so the sieve tubes are part of the phloem, and they're made of cells that are called sieve elements. And the interesting thing is that these living cells, I spelled nucleus wrong. They're living cells, these sieve cells, but they have no nucleus, and they have no ribosomes or tonoplast. Okay, and their diameter is 10 to 15 micrometers. Okay, and the end walls of the sieve plates, of the sieve cells have sieve plates, right? It kind of looks like this colander, and it has large pores that the water and the food can move through. So alongside the sieve tubes are companion cells, and these so mesoville cells in the leaf are close to veins that contain these sieve tubes. So sucrose will travel to the phloem through companion cells in two ways. Okay, so they can go from cell to cell through the plasmodesmata, okay, along cell walls in the mesophyll. And then carrier proteins in the cell surface membranes of companion cells actively pump sucrose into the cytoplasm. And when you see that these companion cells are actively pumping sucrose into the cytoplasm, that should be an indication to you that ATP is being used. Okay, this is active diffusion, not passive. So in the xylem, it's passive diffusion, right? Water is just moving down the concentration gradient, where here sucrose has to be actively pumped in the companion cells. So from here, it passes through the plasmodesmata into a sieve element. The accumulation of sucrose and other solutes, like amino acids in the sieve elements, lowers the water potential, so that water diffuses in by osmosis from adjacent cells and from the xylem. So this creates pressure in the sieve elements, causing the liquid, which is the phloem sap, to flow out of the leaf. Okay, and again, take time to hit pause and to really look at these models. You need to know all of these elements. Okay, any vocabulary word that's on this model, you need to know, and you need to understand these processes. Okay, so phloem sieve elements are adapted for transport as it has end walls that have sieve pores, which allow the sap to flow freely, little cytoplasm to impede the flow, and the plasmodesmata allows assimilates to flow in from companion cells. Okay, so phloem cells versus xylem cells. So sieve elements differ from xylem cells because they are alive. Sieve cells have some cytoplasm with organelles. Sieve cells are not lignified, okay? So they do not need to withstand the same forces that exist in xylem. They are not really woody and obviously are made of living cells, not dead cells. Okay, sucrose is unloaded at sinks. So this is taken up by the cells and is respired or stored as starch. And this reduces the concentration of phloem sap and lowers the pressure. So it helps to maintain a pressure gradient from source to sink so that the sap continues to flow in the phloem. Okay, and so here is a picture from your book. So you can see images A and B here. So A, you can see the phloem sieve element, the companion cell, and the source. Okay, and this is really showing you the process where things are being loaded. Okay, so the assimilants are being loaded into the sieve element, and in this case, again, this is sucrose. So the sucrose in solution is moving from the mesophyll cell, okay, across the leaf to the phloem tissue. And it may move by the symplastic pathway, moving from cell to cell via plasmodesmata, which we just talked about. But it can also move the apoplastic way, which is traveling along the cell walls. At the moment, they don't really know how the sucrose crosses the cell surface membrane to enter into the apoplastic pathway, but it's thought that it probably can as well. It is known that the companion cells and sieve elements work together. So sucrose is loaded into a companion cell or directly into a sieve element by active transport. Okay, hydrogen ions, and if you look at this model, you can see hydrogen ions are pumped out of the companion cell into a cell wall using ATP as an energy source. This creates an excess of hydrogen ions in the apoplastic pathway outside of the companion cell. The hydrogen ions can move back into the cell down their concentration gradient through a protein which acts as a carrier for both hydrogen ions and sucrose at the same time. The sucrose molecules are carried through this co-transporter molecule into the companion cell against the concentration gradient. The sucrose molecules can then move from the companion cell into the sieve tube through the plasmodesmata, which connect them. Okay, and so a similar thing is happening with unloading. So unloading occurs into any tissue which requires sucrose. So any tissue that's requiring food, essentially, or requiring sucrose, whether that's the root or a berry or a flower, right? Anything that has sucrose. So it's probable that sucrose moves out of the phloem into these tissues using both symplastic and apoplastic routes as well. Phloem unloading requires energy, and similar methods to those used for loading are used as loading and unloading. Okay, so but once in the tissue, the sucrose is then converted into something else by enzymes. So decreasing its concentration and maintaining that concentration gradient. And one example of an enzyme is invertase, which we've talked about, which hydrolyzes sucrose back into glucose and fructose. Okay, so it's really important to understand that the xylem has passive transport. Okay, and it's water and dissolved mineral ions moving one way through tissue that is composed of dead cells, whereas the phloem are living cells with these sieve elements that have these sieve plates where sap is moving through. So, right, water and sucrose and amino acids and other various things, and they're moving. And they're moving the same way with this pressure that's driving them. It's this pressure-driven mass flow, okay? And this pressure is created, it's actively created through the use of ATP, okay? So it's this idea that there's these proteins that are transporting both hydrogen and sucrose. And so when the hydrogen is moving down the concentration gradient, it moves through that protein, allowing for sucrose to move up the concentration gradient. So it's at the expense of ATP. Okay, and that happens in this companion cell, which then loads that sucrose into the phloem. And it's the same process. It moves down through the flow of that mass flow of water that's diffused through osmosis from the xylem, and then it moves down to the sink. So whatever cell happens to need sucrose, and then it's unloaded the same way with ATP. Okay, so. Again, xylem, water, phloem, food, and you really need to understand all of the mechanisms and vocabulary in this process. Okay, and we know that osmosis is occurring against down the concentration gradient, but with ATP, we have a movement of sucrose against the concentration gradient as hydrogen ions move down the concentration gradient. Okay, so I want to make sure that you're reviewing all of the basic concepts that we covered in this lecture, but it's really, really important in Chapter 7 especially that you're going back and you're looking at all those pop-out boxes. So all of those boxes in your Chapter 7, you need to memorize those. So that's everything from translocation to just the basic anatomy and structure of transportation in the plant. Okay, it's really important that you understand all of those and that you are reading through the chapter. So thank you very much. Yeah.